تمام، يعطيكم العافية. نبلش المحاضرة الثانية للimmunology. We'll try to cover more tracks than last time. هلا نرجع على هاي الفيجر. This is the central figure for the immune system. هذا figure راح نبني عليه طول الفصل. كل محاضرة راح نضيف على هذا figure something else, something new. So this is your central figure that you need to know. In this figure, you can see that there is a central cell. The central cell is a T cell. It's called a CD4 T cell or a helper T cell. This is the central cell. هاي هي المديرة. This is the cell that receives input. شو المدير بيساوي؟ المدير بيجي الموظفين بيحكوا له boss, we have this so and so and so, and then the المدير شو بيحكي؟ اعملوا كذا وكذا وكذا. So what is the input that the helper T cell receives? The input that it receives is the antigen from the antigen presenting cell. So an antigen presenting cell واحد من هالموظفين بيشتغل بالفيلد بلفلف، لقى foreigner أخده. عادة حكينا it's a macrophage or a dendritic cell. أخده بكسره. Finds the unique part. We mention how راح نحكي عنه هذا القدام. We will mention how that happens. The viral epitope is then presented on a class 2 MHC protein. This is unique for حكينا لدى antigen presenting cells. MHC class 1 is present in all other cells. فهاي بتروح على الهيلبر تي سيل بتحكي لسيدي أنا وصلت لقيت كذا وكذا وكذا. This is what I found. هلا هنا نقطة مهمة إنه the helper T cell, the viral epitope is presented on an MHC class 2 protein to the helper T cell on to its T cell receptor. نحكي عن T cell receptor بمحاضرة كمان. So the T cell receptor and the MHC protein on the APC both have to be specific for this molecule. راح نحكي هذا لقدام كيف that happens. So not all APCs can عفوا that's not let me correct this. All APCs can present on MHC class 2, but not all helper T cells can react to this antigen. فهاي ال APC once it degrades the virus and takes the unique part, بتروح بتدور على مدير اللي بيعرف هاي الشغلة. So not any helper T cell. So it goes to the nearest lymph node مثلا, find lots of helper T cells. It will find one of them that can react. This هاي الوحدة تحكي أنا هاي شغلتي, أنا مدير هاي الشغلة. تعال, yes, let's start working. هلا راح نحكي لقدام كيف how the helper T cell decides إنه I need to go in the interleukin 4 or the humoral pathway إنه أعمل antibodies ولا بدي أروح على cellular pathway, cytotoxic pathway. نحكي لقدام how the helper T cell decides, but it's basically إنه it depends on عادة depends on the protein and the APC. بتجيب لها الفيدباك, بتاخذ feedback من ال APC. تمام. So what does it do? Let's say it decided to go towards the humoral pathway. بتروح and then it will find a B cell that can react to this epitope, and then see this one can react to this epitope, so it will come and activate it. هاي البي cell تتحول لبلازما cell. It's because it can react, its antibody is specific to this virus. It starts to make the antibody to this virus. هاي واحد. Or it will say, no, I need to kill the cells that are infected with this pathway, with this virus. So I need to kill, the virus is inside these cells. I need to kill it. So what do? I need to find, حكينا, every cell in the body shows the protein makeup inside it on its class 1 MHC protein. So I need to find a cytotoxic T cell that is specific for this viral epitope هذا الأسود, and go find cells that are showing that they have this virus inside them and kill them. So it will go to the CD8 cell, it will go and find a cell with this infected with the virus and kill it. So this is what's going on here. هلا كل مرة انت بتلاقي حدا specific for a specific antigen, مثلا حكينا لقينا helper T cell تعرف هذا الانتيجين, او لقينا B cell تعرف هذا الانتيجين, او لقينا cytotoxic T cell تعرف هذا الانتيجين. Every time you find the correct cell, معناتها هاي السيل بالنسبة لي مهمة. هاي السيل راح استخدمها in my environment a lot, because مثلا I come into contact with this virus all the time, I come into contact with this bacteria all the time, with this epitope. So this cell that I made, which seems to be specific to this virus, to this epitope, هاي خلية مهمة. So each and every time there will be clonal proliferation. This one I will make a lot of clones of it. I will make a lot of it. And وهاي إذا استخدمتها نفس الشيء, I will make a lot of it. وهاي إذا استخدمتها راح أعمل منها كتير. And every time I do clonal proliferation, every time I proliferate the cell that I want, I make memory of it. So next time يكون عندي منها جاهز على الشلف. And the next time I meet this virus, I will go not from zero, إنه أقعد على APC وأقعد أدور على حدا مين يا عم مين بيشتغل على هذا الفيروس. No, I found it, I know where it is, I keep it ready for the next time. This is the central dogma of immunology. Just know everything we said here so far, and you will be good. So, cell-mediated immunity, let's go through. When a foreign invader, مثل mycobacteria, enters the body, it is first ingested by macrophage. زي ما حكينا, this is the common first step between cell-mediated, between innate and adaptive immunity. Both of them will have the ingestion of the foreign invader. But the difference here is the bacterium is broken up. It's into, and the fragments are shown, or the epitopes, on the class 2 MHC complex على ال APC. Neutrophil, does it have class 2 MHC complex? No, because a neutrophil is only a phagocyte. It's part of the innate system only. So the antigen, class 2 MHC a protein, interacts with the antigen-specific receptor and on the surface of helper T cells, and helps the activation of the adaptive immune response. تمام. Activation, هاي حكيناها كلمة activation. Each time I find the correct cell, the correct antigen, so an antigen-specific cell, I find it, I activate it. مين الactivator? CD4 cell. CD4 cell activates itself أولا بالinterleukin 2. إذا نرجع للسلايد. So هيو, it first activates itself and then activates others with either interleukin 2 for the cellular part or interleukin 4 and 5 for the humoral part. Each time it activates something, it will proliferate. بيعمل منهم clones. يعني clones. So it's identical copies of the cell. Many copies. Proliferation of, this is clonal proliferation. وين كنا? Okay, here. So helper T cells occur as a result of the production of interleukins. The most important interleukin, إنكم تعرفوه أول واحد وأهم واحد, interleukin 2 is the activator of CD4. So CD4 activates itself and the cellular pathway with interleukin 2. These activated helper T cells, with the aid of the now activated macrophages, start one important component of cellular immunity called delayed hypersensitivity reaction, or type 4 hypersensitivity reaction, رح نحكي عنها قدام, specifically against MTB. So now we want to kill the cells infected with TB. So the cytotoxic or cytolytic T lymphocytes, from CD8 lymphocytes, get a license to kill a specific cell. يعني هي موجودة هي الخلية CD8 cell. It has a receptor. It has a T cell receptor that reacts with this viral epitope, but it cannot kill cells with this viral epitope unless it is given the license to do so. Activated by interleukin 2. حكينا every time you hear the word in immunology activated, that equals شغلتين. الأولى clonal proliferation. الثانية creation of memory. Every time you hear activation, that means again clonal proliferation, and then memory cell production. So these activated helper T cells, with the aid of the new activated macrophage, start start the specific immunity against MTB. So T lymphocytes, they get this license once they are activated to start killing the cells that they are specific to. So if a virus مثلا enters the respiratory tract and infects the cells of the respiratory tract, and viral envelopes glycoproteins appear on the surface من جوا لأنهم they are showing what they have inside. Any production of proteins inside, you are going to present on your MHC1. في عندك pathway to show every protein اللي بيطلع من هالريبوسومات من الاي ار بدك تحطه على ظهر الخلية to show the human body in what am I showing self proteins or am I showing non-self proteins? Are my self proteins correctly folded ولا لا? Are my self proteins working ولا لا? Should I be destroyed or should I continue to live? That's the role of MHC1. هذا أهمية ال MHC1. I am showing what I have, what I'm making. بما إنه الفيروسات بيستخدموا البروتين mechanisms, protein production mechanisms تبعين الخلية، معناته by the same factor راح نحط whatever proteins that we're making that are viral proteins, I will show them on my own class 1 MHC. And this way I will be able to identify which cells are infected by the virus. So a cytotoxic T cell اللي عندها T cell receptor that is again antigen specific to the viral antigen, class 1 MHC protein complex. It is then stimulated, activated clonal proliferation, memory, and grows into a clone of cells with interleukin 2 produced by helper T cell, and then they go and find all the cells that are infected with this virus and kill it. هلا these cytotoxic T cells specifically kill cells infected with this مثلا هون in this example is the influenza virus, and not cells infected by other viruses. So بما انهم they're only specific for this epitope of this virus, they only kill this specific virus. مثلا إذا حتى طلع عندك another influenza virus with a different epitope ما راح تقتل هاي الخلايا. Only the ones with this epitope of this virus. The epitope اللي هو the unique part of the protein that I am presenting on my MHC1, which the APC also presented to the CD4 cell. So by recognizing the viral antigen class 1 MHC protein on the cell surface and releasing the perforins، بيجي كيف تقتل الخلية، تمسكها with T cell receptor، تمسكها بال MHC1 complex تبعها MHC1، تلزقوا مع بعض and then start releasing bullets on it. These are called perforins, bullets. It keeps poking hole in the membrane and then the infected cell will die. Antibody mediated response. Antibody mediated response is the antibody production usually involves the collaboration. هلا يعني بدون ما تطلع انت which 3 cells do you need to make. حكيناهم من الفيجر، حكينا عندنا ال APC بدها تيجي، بدنا CD4 cell عشان to activate, and then we have the B cell which become the plasma cell. So these are the collaboration of the 3 cell types. After processing by an antigen presenting cell, fragments of the surface will be presented on class MHC complex 2, which the protein will be presented to the helper T cell. المديرة بتعرفها، المدير اللي بيعرف هذا specific antigen برد علي بيجي and will activate itself with interleukin 2. So antibody producing pathway will first synthesize interleukin 2 and then interleukin 4 and 5 to activate the B cell. So احنا انتقلنا ل humoral pathway, the antibody producing pathway, not the cellular pathway. So interleukins 4 and 5 activate the B cell and produce specific antibodies to presented antigen. هلا specificity is not mediated by interleukins. هاي كتير مهمة. interleukin 4 and 5 بيستخدموهم كل ال CD4 cells. كلهم بيطلعون the same interleukin 4 and 5. This only is released, this is just هو عبارة عن كلمة. interleukin 4 عبارة عن كلمة means activate يا B cell. That's interleukin 4 and 5. interleukin 2 means activate يا CD4 cell and يا CD8 cell. That's what it means. It's just a word. It's just a word. It's not specific. أي وحدة أي خلية. But who was specific? The MHC2. The T cell receptor was specific. So the T cell receptor of the CD4 cell he can't specific. هي اللي عرفت what to do. The antibody was specific to this specific protein, to this specific antigen of this specific virus مثلا. So specificity هون is not mediated by the word that they say to each other, the interleukins, it's rather by their own receptors that makes them unique. So each T cell receptor, هاي النقطة مهمة, each T cell receptor is unique, each antibody is unique to a specific antigen. كل خلية عندها واحد من التي سيل ريسبتور، نوع من هذا سبيسيفيك، فمعناته عندك ملايين انواع من تي سيل ريسبتورز، كل واحدة على different kind of CD4 cell. And the same happens for B cells. كل واحدة عليها different antibody that reacts to a different antigen، فعندك ملايين أنواع من ال B cells. So you are looking for the specific CD4 cell for this specific epitope to go and find a specific B cell and activate it for this same epitope so it can activate it and start making antibodies. You can realize this is time consuming. عبال ما ألاقي الخلية الصح وألاقي البي cell الخلية الصح، وهذا this is all time consuming, and this is why the adaptive system is time consuming. And this is why I make memory، لأنه متى ما لقيت السبيسيفيك خلية، بما إنه عندي ملايين البي cells وملايين CD4 cells that are each specific to a specific epitope، معناته إذا لقيت اللي حكينا زي ما حكينا، إذا لقيت وحدة بتهمني، وحدة اشتغلت على epitope أنا شفته، معناته هذا epitope موجود in my environment، معناته I need more of this again. So I activate it, I clonal proliferate, and then I make memory مهمة. Even though antibody production usually needs helper T cells to start its production, some antigens can activate B cells directly. This is a second mechanism نرجعها. Some B cells، بما إنه هنا I need a specific helper T cell and then another specific B cell, some B cells can actually present antigen to helper T cells, and then they can activate, some of them can actually activate themselves right away. This is a mechanism to start making antibodies production بسرعة، بدل ما أنا I depend on two different specific cells. Here I can start antibody production in a T cell independent manner. So it's called T cell، اللي هي مين T cell independent، اللي هي ال 4. So the CD4 cell. So T cell independent antigens, some of these antigens can activate B cells. Some antigens، هاي مهمة كلمة، some antigens can activate B cells without the need of T cells. هذولا the T cell independent response is only IgM. راح نحكي عن أنواع ال antibodies. So IgM is the first antibody that you start making, the least specific، يعني the broadest of the antibodies, and it is the first that you start making, and then you switch to IgG, IgA, IgE, and then become more specific and more efficient. So the coordinated response against virus infected cells. So virus infected cell، هاي الvirus infected cell. So this is the T cell مثلا، هو T cell، عفوا B cell T cell independent activation or B cell presentation of B cell to CD4 cells أو T cells هون. So هاي الفيروس طلع من خلية يدمرها، طلع منها. This IgM is specific to this antigen, seems like, and this is an activating antigen, so we can start making antibody. Also حكينا ال B cell can also present antigens. It can also، هاي نقطة نضيفة جديدة نضيفها. It can also process antigens and present them on MHC2. So any cell that has MHC2 is an antigen presenting cell. هلا صرنا عندنا macrophage، dendritic cell، رقم 3 صار عندنا نضيفها B cell. B cell is a APC. So it goes and presents the CD4 cell with the epitope. CD cell، هاي عرفنا أنا معناته هاي CD4 cell مهمة. It knows this epitope, it activates itself, makes more of itself, goes activate the CD8، بروح يقتل بالporphyrins، يقتل الخلية by knowing which cell on MHC1 has this viral epitope. So this T cell receptor is the same as this T cell receptor. It is specific to this epitope, and then the same thing, you can activate this B cell to make antibodies. So the first cycle on the right حكيناها هاي اللي بالصورة اللي قبلها. So the viral epitope plus MHC2 with the viral epitope presented CD4 cell with the helper T cell, and macrophage activated the CD4 cell with the interleukin 1 اللي هو هنا. So the activated CD4 cell produced زي ما حكينا 4 to 5 interleukins activate B cells to produce antibodies. تمام. The second cycle, the B cell will take up the viral antigen using IgM surface antibodies specific to that antigen. هاي الإضافة بهاي الصورة is this basically. فشو هاي الفكرة what's new is B cells can independently bind to antigens. 1. 2. B cells can present this antigen to the CD4 cells and activate themselves. If they do not do this, then they can activate themselves independently if this antigen is correct, independently of the CD4 cell, and can produce IgM only. So that's what we need to know here. So summarize the B cell function. They can recognize antigen using their IgM immunoglobulin. IgM can recognize foreign proteins. هاي نقطة مهمة. IgM receptors can recognize foreign proteins, carbohydrates, lipids, DNA, RNA, other molecules, basically everything. So you can make IgM لكل شيء، معناته you can make an antibody to almost everything. هاي نقطة كتير مهمة وراح قدام راح تنعاد كتير. IgM surface antigen receptor has a remarkable ability to be able to recognize virtually every known molecule, so you can make an antibody to almost everything. They also present antigen epitopes to CD4 helper T cells, and only هاي النقطة رقم 2 M2 using MHC class complex protein, but their MHC2 protein can only present antigen fragments to helper T cells, and here you will see they can only present proteins, they can only present amino acids, so they cannot present. If they can recognize everything else, they can only present, so they can get only their activation if they come into contact with proteins. هذا راح نحكي لقدام how this changes stuff. So eventually they also differentiate to plasma cells. طبعا plasma cell is a factory. مثلا هذا IgM تبعي مهم لأنه أنا لقيت الفيروس، أنا عفوا الابيتوب whatever it is. So I switch myself. بغير الجينات تبعتي بصير I only do nothing. I'm not presenting antigens. I'm not doing anything else apart from pumping out this important antibody. So I keep making a lot of antibodies. That's how you raise your antibody titer جسمك. So the type of immunity that provides resistance before exposure to a harmful substance. هلا صار حنا هذا صرنا نفهم أكتر إنه before exposure it's only an innate type of immunity. So as a preventative type of immunity is not specific. هل في antibody specific؟ هل T cell receptor can specific? There's nothing like that. So these are defenses against many types of molecules. These defenses range from زي ما حكينا keratin layer of the skin, enzymes مثلا lysozyme, neutrophils, complement proteins. These are all does not improve after exposure. ما في شيء memory، ما في شيء clonal proliferation. There's no activation, right? كل هذا بالinnate immunity now that we know it more. So بنرجع لهاي table again, factors that limit entry to the microorganism. Again, all these cells that important components of the innate system اللي هو keratin layer of the skin. This is a mechanical barrier. In your secretions you have lysozyme that degrades peptidoglycan in the bacterial cell walls. Cilia بضلها elevate the mucus to trap organisms. The mucus itself traps microorganisms, and then the cilia will move this mucus down to the stomach to destroy, in the low pH in the stomach, to destroy these organisms or reduce their growth. The low pH in stomach, vagina, and the fatty acid in the skin, all these have microbe killing or retardation ability. Surface phagocytes, alveolar macrophages, neutrophils, whatever, they ingest and destroy microbes. Your defensins رح نحكي عنهم لقدام. Again, something similar to lysozyme, but they are cationic peptides that create pores in microbial proteins, membranes. كمان نضيف normal flora. إيش يعني normal flora? The normal pathogens that are مش pathogens, the normal bacteria that are present on your skin, in your mucous membranes, كلهم هذه موجودة. All of them are occupying space. So because they're occupying certain space, certain niche زي ما حكينا, they are preventing other pathogenic bacteria from colonizing or causing the infection. This is another type of your component of your innate immunity. هلا other parts of your innate immunity that can limit the growth of microorganisms within the body. This is not outside. مش إنه to prevent them coming into the body to within the body. NK cells, these are naturally competent cells that kill virus infected cells without بدون ما يرجعوا لحدا. They have the license to kill right away, and they do so رح نحكي عنه قدام, but without the need of being activated. Neutrophils حكينا, so they are neutrophils, they are phagocytes, but they do not present antigens. Macrophages and dendritic cells حكينا, they have the same ability, but they are also able to present antigens. Interferons are proteins that you can make that inhibit viral replication, inhibit the polymerases and of the virus, the ability to replicate their DNA or RNA. Complement, C3b بيشبه, has a function that has a similar function to antibodies. It can bind foreign objects without being specific. It is specific to them, but I do not make it. يعني مثلا كل البكتيريا عندهم peptidoglycan. I do not need to find a unique structure بالنوع المعين من البكتيريا عشان أعمل منه antibody, مثلا كابسول. But مثلا C3b can opsonize any bacteria basically, زي ما اللايسوزايم بيشتغل على any peptidoglycan. C3b can opsonize, يعني بحضر البكتيريا to be engulfed by macrophage. فهذا this is something similar to antibodies, but the antibodies are made to unique parts to unique organisms. مثلا شيء مش موجود إلا بالستاف أوريوس, أو مش موجود إلا ب influenza virus type H1N1 whatever. Okay. Another thing that the complement do is make a membrane attack complex, which work in a similar fashion to perforins and create holes in bacterial membranes. Transferrin lactoferrin حكيناها بالمحاضرة الماضية تبعت الجروث 2. حكينا إنه حكينا there's a tie-in with immunology that your body wants to prevent bacterial growth, and they require iron for their growth. So you have حكينا we have acute phase proteins, the protein binding and transferring molecules in your body اللي هم transferrin and lactoferrin. So you بتزيدهم هدول انت in your acute phase reaction من الليفر. اذا الليفر بحس انه في من الماكروفيج, when a macrophage is activated تحكي للجسم انه I found something, the liver will start, كل ما بتحكي I found something, كل ما بتحكي I found something معناته there is more and more and more bacteria. The liver will start to make more and more acute phase reactant. One of them is transferrin, which sequesters iron, and binds all the iron in the body to prevent bacterial growth, because iron is required for bacterial growth. Fever is if I come into contact with organisms, we have an innate component of our system to increase our body temperature. حكينا كمان بمحاضرة الجروث الماضية, هلا قبل شوي حكيناها, I just recorded it, but in اللي هي I think growth 2 is حكينا إن البروتينات بحاجة إنها تكون within a specific temperature to be correctly folded and functioning. فإذا بجسمي أنا برفع temperature, I'm trying to misfold and destroy the proteins of microorganisms to destroy their growth or reduce their growth at least. Inflammatory response to limit اللي هو you know it, and then apolipoprotein B RNA editing enzyme, they cause hypermutation in viral DNA and mRNA and then make more errors for them and try to destroy them this way. All of these are part of your innate system. Important components هاي حكيناهم هنا. So this is the same table, I just split it in half. Nothing else here. Okay, the innate arm of the host defense performs two major functions. So they are killing the invading microbes, and they're activating the adaptive immune system. So حكيناها macrophage versus neutrophil. So even though the innate system is able to eliminate microbes and prevent infections, it cannot protect the human body by itself. This is evident by SCID, which is a severe combined immunodeficiency disease. they have intact innate system, but they do not have an adaptive immune system. So obviously they will suffer from repeated life-threatening infections. The way the innate system recognizes a foreign invader from the body's own cells, that several components of the innate system recognize is what is foreign versus not self. By detecting certain carbohydrate or lipids on the surfaces of microorganisms, هذا يسموهم pattern recognition receptors. مثلا كيف يعني الطريقة أنا فاهم الطلاب فيها هاي النقطة، إنه أنا جسمي بستخدم جينات مختلف عن جينات البكتيريا. الباترن أو أنماط البروتينات تبعاتي وكيف أنا I fold them how I make them is different. For human proteins are different from bacterial proteins. They look different from bacteria. There is a pattern بتكرر بالبروتينات تبعين أو الريسبتورز أو whatever تبعين البكتيريا. هذا النمط من تشكيل البروتين مختلف عن النمط اللي عندي. ف جسمنا تدرب، أجسامنا تدربت على معرفة هاي الأنماط، إنه هذا النمط المعين موجود بالبكتيريا، هذا النمط معين موجود بالبكتيريا الفلانية، هذا النمط معين موجود بالبكتيريا أو بالفيروس الفلاني. So by knowing these carbohydrates or lipids on the surface of microorganisms، مثلا ال DNA تبعت البكتيريا تستخدم FMET، ما تستخدم مثل جروب مثلا ميثايونين different kind، ممكن تستخدم FMET. ما هذا الFMET مش موجود إلا بالبكتيريا. معناته إذا شفت FMET أنا معناته في بكتيريا. There is bacteria that is trying to replicate DNA. So this pattern of growth means إنه مثلا so these are called molecular patterns that are pathogen associated molecular patterns PAMPs. And this is present on the surface of many microbes and it's basically very important. You know, it's not present on our cells, on our own human cells. High strategy. So this is a component of innate arm that do not have to have high specificity. فهاي نقطة كتير مهمة. مثلا إذا إشي بعرفه أنا بجسمي إنه I don't have it at all or this pattern I don't use at all، معناته this is not me. So this is not myself. So I can safely make an immune system, an immune response against this. There are two classes of surface receptors. بسموهم toll-like receptors or mannine and mannan binding lectin receptors MBL and TLR, toll-like receptors and MBLs. So these detect non-self cells من هاي النقطة اللي حكيناها من PAMPs from outside the cells. These two classes perform the same function, which is however it is for intracellular invaders. For intracellular invaders, we have the NOD receptors or reg-1 helicase receptors. Mutations in these genes إذا ما عندي أنا toll-like receptors MBLs or NODs, I do not have this ability of recognizing non-self molecules in a non-specific manner, and this allows severe bacterial, viral, and fungal infections. The most important of these هذولا pattern recognition molecules سمعتوا عنهم قبل هيك هم toll-like receptors. This is a family of 10 receptors found mainly on the surfaces of macrophages, dendritic cells, and mast cells. These toll-like receptors recognize حكينا various microbial components, which then they activate transcription factors and promote the production of pro-inflammatory cytokines, and they enhance the immune system. Note that the type of the host defenses mounted by this is depending on the type of the organism that they encounter. تمام. An antibody-mediated response is produced against one type of bacteria, but a cell-mediated response is in response to another type of bacteria. حكينا نتذكر بداية المحاضرة حكينا there's feedback that the CD4 cell can get from the macrophage عشان تعرف هي أنا أوجه حالي بدي أروح أنا على الانترلوكين 4 or 5 pathway ولا بدي أروح على الانترلوكين 2 and cellular pathway، بدي أروح cellular ولا humoral. One such mechanism that helps the decision is these toll-like receptors. فإذا أنا بعرف هذا الtoll-like receptor مش عندي 10 أنواع منهم، إذا هذا النوع إذا أنا if I activated toll-like receptor 3 for example, toll-like receptor 3 is something that is seen on viruses، معناته I require this type of response. مثلا أو إشي داخل الخلية معناته I want cytotoxic response. So the CD4 cell will mount a more cytotoxic response more than antibody response. إذا كان toll-like receptor مثلا for a capsule of a bacteria which requires antibody to be engulfed، معناته أنا toll-like receptor مثلا راح نحكي arbitrarily مثلا 7. So if I meet 7 معناته I most likely I want to go to the antibody response. So this is one such input that can direct the immune response towards a cytotoxic response or a human response. بعدين بكرة راح نغير هاي الأسماء، راح نحكيها TH1 TH2. The determining factor on which type of response is initiated depends on the cytokines produced from the macrophage اللي لقت the one that meets, the one that is in the field that meets. So it depends on which type of receptor was activated. The macrophage is external or internal, and this depends, changes the cytokines that are being produced. So effector cell, this is مثلا macrophage, and found a target, target is non-self, it's recognized as microbial cell, non-self, so right away activate immune response. If I recognize the missing self, this is a target cell, but it's not ما بتعرف حالها إنها self, so there's something wrong with it, so this will activate NK cell and kill it. Or altered cells، إنه مثلا هي خلية بشرية بس غيرت حالها شوية، so there's something wrong with it again, and then this will tell it to go to apoptosis and kill itself. These is types of these decisions that can be made. شو في عنا أنواع؟ كنا نحكي toll-like receptor 3 و 7. So let's talk about actual responses. Endotoxin بال LPS حكينا عنه on the surface of gram-negative bacteria. The lipid A portion حكينا هذا البورشن إنه إذا كان موجود will cause septic shock. So when released from bacteria, LPS combines with LPS binding protein. This is a normal component of plasma. نسميه إحنا LPS binding protein لأنه binds LPS obviously, and this binding protein transfers the LPS to a receptor على الماكروفاج اسمه CD14. So now I know عندي صار input إنه هذا I'm meeting LPS. LPS requires مثلا an antibody response. So LPS stimulates a pattern recognition receptor أو toll-like receptor 4 اللي هو transmits signal via intermediates to the nucleus تبعت الماكروفاج، بتحكي إنه أنا مثلا I want to make a TH2 response أو human response or antibody response. So gram-positive bacteria and yeast are recognized by toll-like receptor 2 مثلا، مش 4 زي موجود بالgram negatives. So this induces the production of cytokines إذا كان 4 مثلا interleukin 1, 6 and 8 and tumor necrosis factor, which are specific to help the destruction of this bacteria. راح نبلش نحكي عن co-stimulator signal قدام to activate the helper T cell and then modify response and reduce the immune response to prevent septic shock or try to limit the bacteria before it can cause septic shock. Other examples of pattern recognition molecules هم MBLs, mannan binding lectins. Many bacteria and yeast have this polysaccharide called mannan. بما إنه bacteria and yeast هم مش human cells, so if they have the mannan, so if I have a mannan binding lectin, I can this is a pattern that I don't have, so a mannan binding lectin binds to the mannan on the surface of these microbes and activates the complement system, which is an innate part of your system. So the activation of the complement system enhances phagocytosis, and then this improves your innate system. MBL is part of your normal serum proteins, and زي ما حكينا is increased in the acute phase response, which is the response اللي حكينا when macrophages meet a foreign body, they start declaring to the body I met somebody foreign, and the liver starts to react the more it hears this signal and release these acute phase proteins. حكينا الأول كان transferrin، الثاني هلا صرنا نعرفه اللي هو MBL, which are enhancing and activating of complement. Other examples of pattern recognition self against non-self هم حكيناهم ال NOD receptors, part of the cell. This is recognized. These are intracellular receptors. معناته I'm moved towards intracellular bacteria. معناته more cytotoxic response. Reg1 helicase, also nucleic acid of viruses, مش bacteria, and cytoplasm in the cytoplasm of infected cells. لهون بنوقف ونخلصنا المحاضرة الأولى, or the introduction of immunology, Again, like I mentioned, the majority of the remaining part of immunology is just basically building on this introductory lecture. فmake sure you know it very well. يعطيكم العافية.